All right, welcome to This Explainer. Today, we are diving headfirst into the ultimate microscopic mystery, the science of bacterial identification. And more specifically, we're going to look at how genetic science has absolutely revolutionized this entire field. We're talking about a massive shift, moving from old-school magnifying glass detective work to cutting-edge molecular forensics, all to figure out exactly what microscopic suspects are lurking in our environment, our food, and, well, our bodies. So let's start with the most foundational question, the one that launches literally every high-stakes microscopic forensic investigation. What bacterium is this? Now I know, it sounds incredibly simple, right? But honestly, whether you're staring at a clinical sample from a sick patient, testing some questionable drinking water, or tracking down a major foodborne outbreak, answering this one single question accurately is, no joke, a matter of life and death. You just can't treat a stubborn infection or halt an outbreak until you know exactly who your microscopic culprit is. Back in the day, to catch our microbial suspect, we would consult the ultimate detective's rulebook, Burgess' Manual of Determinative Bacteriology. This hefty manual is basically the cornerstone of traditional bacterial identification. But here's the thing, instead of looking at a bug's DNA, it focuses entirely on the what. Like, what does this bacterium look like? What does it do? It relies heavily on phenotypic data. We're talking observable, measurable characteristics. Things like cell shape, oxygen needs, basic biochemical reactions, just using those clues to slowly move toward a probable ID. Picture this, a sample arrives in the lab. First, we have to meticulously isolate the suspect. We observe it under a microscope, we stain it to reveal its physical structure, and then we culture it on specialized media, stuff like blood agar or Mac-Hunky agar, just to get a pure culture of one single bacterial species. And finally, we run a battery of physiological tests. It's a very classic, methodical process of elimination. And here's where things get colorful. A simple gram stain gives us our very first major clue, immediately splitting our suspect pool right down the middle, based purely on their cell wall structure. Because of the varying thickness of their peptidoglycan layer, which is just a fancy word for their cell wall makeup, bacteria react differently to the stain. They'll either turn purple or pink. This early split is absolutely critical. It narrows down the potential suspects based solely on structural morphology before we even think about targeted testing. Okay, next up, we build a behavioral profile using biochemical interrogation. We basically expose our suspects to chemicals to see if they produce specific enzymes. So, for example, if we're dealing with gram-positive cocci, a catalase test helps us differentiate between Staphylococcus and Streptococcus. Then, the coagulase test hones right in on that notorious pathogen Staphylococcus aureus. Meanwhile, something like the indole test checks for the production of indole from tryptophan, which is basically a classic calling card for E. coli. But here is the catch, and it's a big one. These traditional old-school gumshoe techniques have some hard limits. Phenotypic identification relies entirely on how a bacterium behaves in a controlled lab. But what if environmental conditions cause those physical traits to change? Or what if two closely related species share almost identical characteristics? Even worse, a lot of bacteria simply refuse to grow on our lab culture plates at all. They're virtually invisible to these traditional methods. So when we rely purely on physical traits, misidentification isn't just a tiny possibility, it is a real dangerous risk. And that is exactly where the science of genetic identification steps in to save the day. We are pivoting from phenotypic identification, which, remember, relies on observable physical traits, over to genotypic identification, where we actually read the exact molecular and genetic sequences of the organism. Instead of asking what a bacterium looks like, we're decoding its fundamental DNA blueprint. Think of it like this. It's the difference between identifying a suspect by the color of their jacket versus identifying them by their actual unique fingerprints. Now, this whole genetic revolution meant we needed a brand new rulebook, Burgess' Manual of Systematic Bacteriology. While that older manual just asked, what is this massive new reference asks, how is this microorganism classified, and how is it evolutionarily related to others? It dives deep into taxonomy and phylology, relying heavily on modern genotypic characteristics and molecular data to place that specific bacterium precisely on the tree of life. To do this, we've got to open up our genetic CSI toolkit, and we're bringing out the heavy molecular artillery here. We frequently rely on 16S-rRNA gene sequencing. Why? Because that specific gene has a very slow mutation rate, along with both conserved and highly variable regions of DNA.
That makes it the perfect molecular clock for identifying species down to their exact lineage. We also use PCR, polymerase chain reaction, to hunt for and amplify specific DNA sequences, which is great for rapidly targeting virulence or antibiotic resistance genes. And for the whole shebang, whole genome sequencing gives us exhaustive data about the entire genetic makeup of the bug.
So let's actually trace how this goes down in the lab using that 16S rRNA workflow. Step 1. We extract the DNA directly from the bacterial cells. Step 2, using PCR amplification, we make millions of copies of that specific 16S rRNA gene, just so we have enough genetic material to actually work with. Step 3, we read the exact genetic code through DNA sequencing. And finally, step 4, we take that sequenced code and run a massive database analysis. We compare our suspect's code against vast global libraries of known bacteria. It gives us absolute forensic clarity that traditional culturing just cannot match. But wait, what if you need an answer, like right now? Well, for ultra-high-speed identification, scientists can turn to MALDI-TOF mass spectrometry. This is wild. We take a bacterial colony, prep it, and literally zap it with a laser to ionize the sample. This creates a unique protein mass spectrum, which is essentially a molecular barcode based strictly on the bacterium's specific protein makeup. We then compare that barcode to a massive database, giving us a highly accurate ID in a matter of minutes. Minutes, not days. If we look at how these two errors of identification stack up against each other, phenotypic ID is definitely highly accessible and generally lower in cost. But as you can see, the speed and resolution are pretty variable. On the molecular side, modern labs are increasingly willing to pay a premium for genotypic ID. Yeah, the initial equipment and per test costs might be higher, but the speed is often incredibly fast. And more importantly, the taxonomic resolution, meaning the sheer accuracy of knowing exactly what specific strain you're dealing with, is just completely unmatched. Now, I want to pump the brakes for just a second. Even with all these advanced lasers and genetic fingerprinting tools, a really vital principle in microbiology remains true. No single test should always be considered sufficient for bacterial identification. You simply can't just blindly trust a machine. Reliable identification requires synthesizing all your data. You have to combine those foundational, old-school observations, like colony morphology and the biochemical profile, with the modern molecular evidence to get a truly reliable 100% confirmed ID. So why go through all this rigorous testing? Well, this isn't just some academic exercise. This science is the absolute backbone of the One Health framework. We use this powerful combination of genetic and phenotypic tools to diagnose infectious veterinary diseases, track deadly pathogens like salmonella in our global food supply, and investigate zoonotic diseases that jump from animals over to humans. On top of all that, by reading the DNA, we can actually predict and monitor antimicrobial resistance before a pathogen even has the chance to infect a patient. It is a total game changer. To wrap up this explainer, I want to leave you with this thought. We've seen how genetic science has given us these unparalleled tools to read the blueprints of our microscopic suspects. But bacteria, they are the ultimate escape artists. They are constantly mutating, swapping genes, and evolving their DNA. So as they change the varied genetic sequences that we rely on to identify them, how must our molecular detective tools adapt to stay one step ahead? It's this continuous, fascinating evolutionary arms race. And honestly, the science of genetic identification is only going to get more incredible from here.